I would like to present Professor Horstmeyer, who is an assistant professor at the Biomedical Engineering Department, and he has a secondary appointment in the Electrical Engineering and Computer Engineering Department. He mostly works with optics, machine learning, and algorithms. Prior to arriving to the Duke University, he was an Einstein postdoc fellow at Charité Medical School in Berlin, where he worked with Professor Benjamin Jukiewicz to apply digital optical phase conjugation in neuroscience. He finished his master's degree at Massachusetts Institute of Technology in 2011 and a bachelor degree in Duke University in 2006. So, Roarke, the floor is yours.
Okay, great, thanks. Hi, everyone. Let me share my screen. Okay, I hope everyone can see my slides. So yeah, I'll just jump into it. So yeah, just a little bit of background about this talk. This was something that's kind of been going on in the background, I guess, and now more the foreground of my research for the last 5 or I think 6 years now. So it's really been kind of a long process of slowly optimizing the technology I'm going to explain. So in 2017, I was just out of my PhD, and my friend Mark Harfouche, who's the co-founder of Ramona Optics, which is commercializing this technology, and I had this idea to kind of push forward on packing cameras close together for applications in microscopy. So that work started, and then I became a postdoc at Charité, and I also worked at the Jean-Paul Aymon Foundation, which is in Lisbon. And so Benjamin Jukiewicz and Mike Orger were involved in this project earlier on, and then I brought all of this to Duke, where there's a number of collaborators. So I guess I'm putting this up first to say that there's a lot of people involved in this project. It has a kind of a long ongoing history. So it's not all me. And so even though my picture is down here, so this is one of the earlier pictures we took with one of our multi-camera array microscopes. There's many others. So what's the kind of motivation for this technology of an array of compact cameras capturing microscopic image details? The original motivation for this was really focused on capturing high resolution video of freely moving model organisms. So in biology, some of you are probably aware that there's these central organisms that researchers cluster around because a lot is already known and people can commonly develop different lines of these organisms, for example, for their research applications. The fruit fly, the zebrafish in the middle, and C. elegans on the right are examples of these. You can see their fluorescent images here are achieved by genetically modifying these organisms to cause them to emit fluorescent light from certain cells, which allows biologists to really understand their detailed behavior and other activity at the cellular level. To observe these types of dynamic moving organisms, you need high imaging speed. Ideally, you want high resolution to see cellular level detail. And so the combination of those two requirements generally means you either need to constrain the organism so it doesn't move around very much while you're trying to study it. And that includes either parallelizing it or embedding it in some gel, or altering, you can give it certain chemicals to have it be like less dynamic or moving more slowly. All of those alter their behavior. So if you're trying to image these model organisms to examine their behavior, those approaches aren't very fruitful. And so the current technology to image moving organisms relies mostly on tracking. So having a rapid feedback mechanism between the slide that's holding the specimen, or actually moving the microscope itself to try to follow the organism as it's moving around. That is a tough task. It doesn't work all that well sometimes. Sometimes it works well, but it doesn't allow you to see more than one organism, right? You can only track one at a time. And so to see multiple organisms that are freely moving around at high resolution, you also need a large field of view, right? A microscope that can see a large area. And ideally, they're moving three-dimensionally, right? So we should be able to extract some sort of 3D information from the scene of interest. And so it's a long list of required specifications, but we set out to try to tackle them. And I'd say we've been successful, pretty successful thus far. And so what kind of optical system can achieve all of these specifications? A single microscope objective cannot achieve these specifications, or a standard single microscope objective. So in this table is a list of kind of regular, I would say, a kind of standard line microscope objective lenses. These are from Olympus, but other vendors have similar specifications. Going down the list, the magnification is increasing, so you're zooming in more, and you can see the resolution increases of these objective lenses. But if you've ever used a microscope, you note that when you zoom in more, the field of view shrinks, right? So the third column here is the field of view of each objective lens, and you can see it drops dramatically as you zoom in more and more, not surprisingly. And so the division of the field of view by the resolution gives you your total number of resolved pixels by the lens, right, independent of the sensor you use. And that hovers around tens, up to 50 megapixels, being a little generous, but generally is much less. So really the limit is the throughput in terms of the number of resolved pixels of the microscope objective lens. That limits the resolution you're able to get over a certain area. So there's been efforts to break that paradigm optically. One notable example is in lithography, where instead of imaging, one wants to project high resolution detail over a large area in the photolithographic process. So this is a picture of Mark and myself next to a lithography lens. You can see the base of it here is down towards our knees. So it's about half of our height. They wouldn't tell us how much it costs, but I know it has dozens of optical elements in there. And this lens can resolve about a billion pixels, or a gigapixel, right? Which is the regime we're trying to operate in. On the right here is some other lenses that I had in my lab that similarly, aren't quite this big, but can resolve maybe 500 million pixels or 500 megapixels. So it really is a tough scaling law that you're up against. And then even if you were to utilize one of these lenses to achieve this goal of seeing these organisms moving around freely over a large area, you would still need a sensor with a billion pixels, right? A billion individual light sensitive elements. And that doesn't exist yet, but perhaps it will in the future. So instead of going for the scale up approach of just make a bigger microscope, we took a parallelized approach. We said, well, we can shrink the microscope into a small package. On the left is a schematic of an analog microscope, a stereoscope you might put your eyes up to view through. If you remove the requirement that a human has to see the actual image, you can make a digital microscope that's much smaller in a small package. It's not too challenging. So we can shrink them down to something like a centimeter cubed, which is kind of the volume we're at now. And then we pack them in an array. Here I'm showing 24 in a 6x4 array, but really this number you can put in is flexible. And each microscope will see a unique patch of the area of interest. So camera 1 will just view this area at high resolution. Camera 2 will view this area at high resolution, et cetera, et cetera. If we want to create an image at high resolution of the entire specimen area, this large petri dish, let's say, we stitch the images together. And the resulting composite is our large area, high resolution image. And so if these micro cameras, we call them, are synchronized, then we have a video at high resolution over a large area. So that's the principle of our multi-camera array microscope. And this idea of using multiple apertures or multiple individual cameras to image is certainly not new, but it's pretty new in microscopy. And I'll explain why that's the case in a couple minutes. But going back, almost 20 years now, there was efforts to use large arrays of cameras to capture what are called light fields. So these are images from many different perspectives of a particular scene of interest that then can be manipulated digitally in post-processing to bring different things into focus, for example. That idea was miniaturized early on by a company called named Pelican Imaging, which I think became a company called Light. I forget the details, but essentially this idea of packing many little cameras together onto a single or a large CMOS chip was motivated, I think, in part by this light field type imaging. But the natural progression of these two led to what we have today, which is really, everyone takes for granted now, but we just have a few cameras on our smartphone, and we kind of take that for granted. The application of multiple cameras isn't just for consumer photography, it's used in astronomy as well. So these are these large area kind of monitoring systems that even amateurs now are putting together to kind of have a pervasive view of the night sky at lower resolution, but over a huge swath to, for example, watch for supernovae or watch, satellites going by, things like that. In the separate kind of technical space, there's also been a lot of development putting lots of cameras together in what are called multiscale imaging systems. So this term multiscale means you have a first optical element that takes light and forms an image of it, typically on a curved surface. And then you have a lot of cameras capturing that image, the kind of shrunk down image or enlarged image. So it's kind of a cascaded imaging design. There was a lot of effort here at Duke about 10 years ago to create this gigapixel camera that was based on this multi-scale design. And then a little bit more recently, there's been a couple efforts to implement a similar idea in a microscopic imaging scenario at high resolution, right? You can image attempt to image a large area. You still need a big objective lens like I was showing, or a giant lens. The intermediate image is formed, and then the camera array captures that intermediate image. So really this talk is focused on implementing microscopic imaging, right? High resolution imaging, but just with a direct camera array, right? So just no additional optics. Maybe towards the end, I'll get into some additional optics, but for the most part, we're just focused on directly imaging. And so there's really three regimes of multi-aperture microscopy, this type of direct imaging at high resolution. So the first is where you are actually treating each individual microcamera as a microscope. So if each camera is zooming in, right, to see at high resolution detail, which usually requires or does require a magnification greater than 1, then no matter how you pack together the microscopes on a flat surface, you'll be left with a gap between the fields of view, right? So that example of observing that large petri dish and stitching the images together isn't possible if you're zooming in with a magnification greater than 1 because of these gaps, right? You'll naturally have gaps and you can't stitch the images together. And so to image across a continuous area, you need to satisfy this condition that the magnification has to be at least 1 or less, right? You need to zoom out a bit, right? And so historically, having a magnification of 1 means you're not really imaging at microscopic resolution. And the reason why is because the size of pixels in most camera sensors, we're on the order of 5 micrometers or 10 micrometers or 15 micrometers, even if you were using a CCD. And so when you image at a magnification of 1, then you're going to be pixel limited to something like 5 or 10 or 15 micrometers, right? Which isn't great resolution at all. And so to implement our microcamera array microscopes, we use newer sensors with very small pixels. So 1 micrometer pixels or even less. We have some with 0.8 micrometer pixels. And so that's newer technology. It's driven by the cell phone camera market primarily. And these smaller pixels allow us to get, micron level resolution, even though we're imaging at a magnification of 1 to see this continuous area. And so this first regime where you're zoomed in, I'm highlighting by this blue line, where you have what we call a tiled imaging configuration, where you see different tiles of the sample of interest, and then you have to scan to sweep out the whole area. The continuous field of view is this green line. You can see the fields of view are just touching at this green line. And that allows us to image a continuous area, right, without any scanning, and record video at that specified resolution. And then there's a third regime. If we zoom out even more and image at slightly lower resolution, then we enter what's called the Multiview Imaging regime. And so you can see here, if I pick a point here on this purple line, this point is seen both by this camera on the left and by this camera next to it, right? So in this purple regime, each point in the scene is captured by more than 1 camera. Here, that just 2, but in general, you can capture more and more as you go more into this multi-view regime. The natural extension of the multi-view regime is as you go towards infinity, then every point in the scene is seen by each camera from a slightly unique perspective, and that's a light field, right? Light field imaging kind of configuration. And so in a paper, I didn't put up the details, but a paper from just a couple months ago, we published a little bit of detail about each of these regimes. You can pick a certain pixel size and plot the resolution you're able to achieve, pixel limited resolution, assuming you can design optical elements to meet this specified resolution, which really isn't that hard because the lenses we're creating are so small. You can generally create pretty high performance lenses. And you can plot the resolution as a function of the magnification and see these different regimes. So the multi-view regime is more on the left here. And so, your resolution in that regime, if you're assuming, let's say, a pixel size of 1.1 micrometers, right, is on the order of here, let's say 20, 30 microns, right? It's not super high resolution. In the continuous regime, we're getting more towards, 10 microns. Now we're closer to 5 micrometers in the continuous regime. And then as we zoom in this tiled configuration, the blue line, you can achieve kind of an arbitrarily high resolution. You can go down to, we're going down to something like 0.4 micrometers, 0.5 micrometers with NA, objective lens NAs, right, that are more like a real high resolution microscope. And so I'll talk about each of these 3 regimes, show some examples, but that's really the technical overview of our multi-camera array microscopes. Our first design looked like this on the left. We have an array of 8 by 12 microcameras here, forming a 96 camera array. Each snapshot captured by this first prototype captured 1 gigapixel. So each camera had about 10 megapixels. And so 10 times 96 is just about a gigapixel. The field of view was large. It was something like the size of a sheet of paper. And the resolution wasn't super great, but decent enough, 9 micrometers per pixel. So an 18 micrometer full pitch resolution. And yeah, we used this to image zebrafish larvae as they were freely moving around a large area. Here, I think something like 100 zebrafish larvae are in this full field of view. We can use tracking software to find and follow each of the unique larvae. There's naturally a lot of data coming off of these microscopes, and so we rely on this type of processing heavily to reduce the total amount of data that we need to save, right? So we don't necessarily need to save any of these gray pixels. We just want to save the pixels that are around each fish as they are naturally moving around. This is the type of detail we're able to see if we zoom in, to each of these heads. But we can see this type of detail, right? This resolution over this entire area, right? So we are capturing this type of detail synchronously over this whole view. And then we actually used overlap between the image sensors to track their movement in 3 dimensions. And so I'll talk more about this in a couple minutes. So that first gen system had a number of limitations. Let's go back. I think I listed. Oh no, I don't. So the main limitation of that first system was that it was slow. We could record at about 1 frame per second with that first camera array. So we spent a lot of time redeveloping the electronics that support our multi-camera array to unlock high-speed synchronized video. And so essentially what we did is we have an array of sensors. Now in this design, there's 54 sensors. We packed everything closer together. So now the field of view is something like the size of an index card. It's an 8 by 12 centimeter field of view, which we chose for a particular reason. I'll show, I think, on the next slide. We get a slightly higher resolution. We get much higher frame rate. So we can go at 10 frames per second when we're imaging at full resolution, which is 700 megapixels per frame, slightly more than that. And then we can go up to 120. Well, now actually we can go up to 280 frames per second. If we reduce the resolution a little bit, we bin the pixels and sacrifice our resolution to get a higher frame rate. And that's because we're really limited by the total amount of data coming off of our microcamera array. Currently, we keep it around 6 or 7 gigabytes per second. We can go higher than that, actually, but just to avoid kind of challenging computational issues, or we generally keep it around 6 or 7 gigabytes per second, which is a lot of data. So you can imagine streaming a video at 7 gigabytes per second, right? If you let that video record for a few minutes, right, you're going to fill up your terabyte hard drive pretty quickly. And so we have a lot of strategies to help deal with that, some of which I'll talk about shortly. Yeah, it's a high data rate endeavor we're working towards here. And so with this updated version 2 design, we really the company that I mentioned at the beginning of the talk, Ramona Optics, is really commercialized this system to look at multi-well plates. So a multi-well plate, for those of you who aren't aware, is a very common item in a biology lab. It's an 8 by 12 centimeter dish that contains many individual wells. Here, this is a 96 well plate. They make 24 well plates. They make 384 well plates, all different sizes, right, going up by factors of 4 effectively, in smaller and smaller wells. And they're all 8 by 12 centimeters, and they're for parallelized experimentation in biology. And so you can put, or many labs put, 1 zebrafish or a couple zebrafish larvae in each individual well and perturb them slightly differently. This is in the background showing a startle response assay, where each small fish is perhaps given a different drug or might be slightly genetically different. And then you can use acoustic stimuli to startle them. So they move rapidly when startled. And recording that and tracking that can give insights into all sorts of things. Like, for example, developing drugs for hearing loss, right? If the hearing system is impaired in these fish and different drugs are being tested, you can see how well are they responding, which is a correlate to how well is their hearing system performing, auditory system rather. So we're recording at high resolution over this large area. So I forgot to mention, we have this tracking software to follow these fish as they freely move, these small larvae. And so we can record a lot of interesting information. Here you can see a couple little red dots. Those are its eyes, and we can watch how its eyes tilt inward and outward as it's trying to hunt. You can see other small things moving around in the dish. That's actually paramecia. Those are things that zebrafish larvae feed on. And so really what we do to deal with this large amount of data most often is we're recording at 7 gigabytes per second across this large 8 by 12 centimeter area at this high resolution. But we're not really directly saving the video even often. We're just automatically computing these statistics, right? This positional information of its body, its tail curvature, its eye orientation angle, and plotting that and saving that resulting data. And so really, we've created a gigapixel-scale microscope, but we've learned quickly that it's really helpful to distill that data into these actionable insights as fast as possible to make both the researchers' lives easier and our lives easier as systems engineers trying to design these data-intensive devices. So this system is also capable of fluorescence. This is an example of an interesting specimen called a xenobot. Again, just zooming into different scales to show the type of video data that the system is able to acquire. Here, again, we're imaging this continuous area and stitching it all together. But this is the type of kind of resolution and field of view our system is able to achieve. Naturally, this is used also in the well plate scenario. Fluorescence is important naturally for these high throughput experiments. A common example is to look at, the video is not playing, but this video shows the heartbeat of this small zebrafish larvae. So we can screen for different heartbeat assays and things like that with this fluorescence capability. Okay, so this that's the configuration where we image across a continuous area at high resolution. The next one I'll describe is more about the multiview imaging geometry. So this is slightly zooming out. So the previous results I showed, we configured the multi-camera array microscope to image at a plane drawn as such, where there's a little bit of overlap between each of the fields of view of each individual microcamera. And we use that overlap to help us with our stitching software. In the multi-view geometry, we're going to zoom out more. And so we can slightly refocus the lenses to image, for example, at this plane where there's 50% overlap, or we can slightly configure them to zoom out a little bit more to image at this plane so that there's 67% overlap. So really it's a design choice where we set this multi-view imaging. But effectively, we want to ensure that each point in the scene goes to at least 2 of the microcameras, or the large majority of the scene. And so if we're able to achieve this multi-view configuration, the effort is to use 3D stereo vision type software, which I'll explain in a second, to capture 3D information about the scene of interest. And so this is what our simple setup looks like. We have our array, we put our samples on the stage here, and then we're illuminating it just with diffuse light, primarily in a reflection geometry. Or we can also illuminate from below for the zebrafish larvae we're imaging in transmission. And we recently published these results in Nature Photonics just, I think, a couple of months ago. And so our system was configured to overlap the micro cameras shown as kind of in this heat map. At the very edge, we don't have overlap. So there's a kind of missing amount at the very edge of our field of view of overlapping data. But as we go more towards the center, we're overlapping more and more. So somewhere on the order of 2 or 3 or 4 views are obtained for each point in the scene, as I'll show. And so this is another view of this overlap redundancy, how we're configuring the cameras to see somewhere between 2 to 4, and then in some areas, 9 views. And that's simply due to kind of our non-imperfect lens alignment, and also just the way we desire to configure it. I think more effort can go into carefully selecting an overlap regime where the numbers of cameras seeing each point is more uniform. But we simply calibrate that, and then we can pass all of the images into some software that stitch them together. So this is an example of a composite of butterfly wings. And while we're stitching the images together, we're jointly estimating the 3-dimensional surface profile, or the height of the scene of interest. And so that process naturally is a coupled process. So if we want to stitch the images together accurately for a 3-dimensional scene, we need to know something about the 3-dimensional height, because if something's much higher, then the stitching won't be accurate as compared to if it's lower at another plane. And so it's a coupled problem, right? Finding the best stitched image and the 3D height of the surface. And so that coupled problem generally has been referred to, or is referred to, as photogrammetry in kind of more long distance imaging scenarios. So what's a little bit unique in our design for a microscope is that we have lenses that are non-telecentric. And so what does that mean? Well, it means that across our field of view, we see slightly different views of objects. So kind of similar to our eyes, we observe parallax distortion as we move around. And so here is just a cartoon of 3 different objects, each at a different height, and an example image that our sensor would capture, our lens and sensor. In a standard microscope, you would actually observe something more akin to what's on the right, where this parallax distortion is minimized. And the reason behind that is just, I guess, driven by the kind of standard design efforts surrounding microscope lenses to achieve this uniformity across the field of view. Has jointly resulted in the minimization of allowing parallax distortion to enter into the lens system. For us, the system on the left is actually way better at giving us 3D information, right? Not surprisingly, if you look at this picture, this parallax distortion is exactly what we need to measure the 3D height of our samples. And so I guess what I'm saying is, standard microscopes, the technology we developed to use this photogrammetric multi-view imaging, it won't work on standard microscopes. It really is uniquely suited to our multi-camera microscope design, where we are using non-telecentric lenses that are just zoomed in to image at high resolution. And so essentially, I'll just kind of go quickly through this. More details about this process are found in this paper in the lower right, a CVPR paper which Kevin wrote. But really, what we're doing is we're capturing an image of a scene from slightly different perspectives. And so here, the vanishing point at the center of this image is redrawn here on the image on the left, which is from a adjacent microcamera, and then same. A microcamera on the left would observe something like this on the bottom, again, showing the same vanishing point. And you can see just visually that the appearance of the blue tower, for example, really elongates over here. And we can observe just by eye and via our own mental processing that this blue tower is higher than the red tower and the green tower, right? But essentially, we've developed software to examine these shifting objects due to that are shifting due to non-telecentricity and stitch them together accurately. And so the process of that is called orthorectification. We essentially measure the shift of each pixel in the scene with respect to a vanishing point. We then rectify them. We shift everything along these yellow vectors and then stitch everything together. The amount that we have to shift each pixel is given by, the magnitude of the yellow vector is directly proportional to the height of the object, right? So essentially, we need to find each yellow vector. That tells us the height of each pixel. That tells us how to shift them correctly to then stitch the final result together. Okay, so that's just summarizing essentially what I said. And so that process, what kind of accuracy do we get with that process? Surprisingly, we get pretty high accuracy for our 3D height. So here's an example of gauge blocks. So these are precisely machined metal blocks, each at a different height. You can see on the left, we're increasing the height from 1 millimeter to 1.02 millimeter, or a 20 micron change in height. Then, up another 30, and then up to 50. So we're kind of going up in increments of tens of microns up to 400 microns. And you can assess the resolution we're able to get just visually. We can't quite get 20 microns, right? These look pretty similar. If you sum up all the pixels, then you do see a shift, but just kind of hand wavy. We do see a shift, arguably, I would say visually from 1 millimeter to 1.1 millimeters. So 100 micron shift. So the height resolution of this technique is, somewhere in the ballpark of 50 to 100 micrometers. And again, we've improved on this. This is kind of earlier results, but that's kind of where it is. And so we're able to use this to capture these large area composites, right? So this is of a printed circuit board. The spatial resolution is something like 10 micrometers, 15 micrometers per pixel. Like I said, the axial resolution is something like 50 micrometers. And we can create these 3D composites of this type of three-dimensional object. And then naturally, we're going back to our motivation, really interested in watching freely moving model organisms. So this is the same technique applied to ants that are freely moving, carpenter ants, at something like, I think this is either 60 or 120 frames per second. I think this one's 60. And so we can see really fine details of these ants freely moving around, but this is, jointly captured the height map of those ants. And so we can create these kind of interesting looking composites of approximate 3D height of the ants as they're freely moving around, but during these interactions and different behaviors. And so from that type of data, we can compute all sorts of interesting statistics. We can examine how their limbs are moving up and down, right? So this periodic movement of the red and blue traces are their left and right forelimbs, mid limbs, and hind limbs. And so the axial movements on the right and spatial movement and X and Y are on the left. We can observe similar effects in zebrafish. So this is a video of the freely swimming zebrafish larvae. We're tracking each larvae and then jointly showing this color view is the height map. And so you can see, I'll just pause it. The zebrafish larvae, not surprisingly, tend to swim either towards the bottom, where they're blue, or towards the surface, where they're red. And they transition from blue to red when they're approaching food. And so you can see a lot of little red dots here. Those are all food particles that are on the surface, floating food particles. And so what we're using this system to do is observe their hunting behavior, or feeding behavior rather, also hunting for fixed or living food, paramecia, for example. But we're watching this transition behavior from low to high swimming as they approach their food to eat it. And so a summary of that is shown here, where a zebrafish larvae, kind of focusing in this middle panel, is approaching a food particle, this little red dot, and we can observe how its body moves upward, tilting, how its eye angles converge, and when it eats. And same within this lower case, eating slightly later. And aggregating those statistics, we can see, we can plot the elevation angle of the zebrafish as a function of its height in the water to observe how they prefer to tilt as they approach their food. And yeah, we're really just kind of scraping the surface of examining how this 3D information can be helpful for our kind of analysis of these behaviors. Okay, so on to the third regime. I'll kind of quickly go through this. We can also zoom in, right, and image at very high resolution. And then we don't have overlap between our individual microcameras. And so this regime really is more like individual microscopes, each zooming in. And then what we do is we can scan the sample laterally and axially to get a kind of three-dimensional scan of the object. So this is really like a parallelized microscope, where we simply have lots of microscopes looking down and we use them to speed up our scanning process. And so it's useful for a large number of applications. We're looking at large histology sections, like large brain slices. We can image 3 slides, entire slides, all at once in 2 minutes. We look at full culture plates. We're also using this naturally to look at the well plates at high resolution. Just showing a couple examples of this. I'll just kind of rapidly go through this. We've designed lenses. This is a 0.3 NA objective lens with a 2.2x magnification that achieves, I think 800 micrometer resolution, 700, 800 micrometer resolution for each of our micro cameras. It fits in an 8.5 millimeter diameter package. So it's really small. It's like the size of a pen cap. We also have now 0.5 numerical aperture lenses that are the same. So we really can go to pretty high resolution, right? Not quite, we haven't done oil immersion yet, but pretty much whatever resolution is needed, we can pack into these little lenses. And then we put them in an array. This array is the size of a credit card. So even smaller, we keep getting smaller and smaller, I guess. But yeah, we can scan out whole slides. Like I said, the system looks something like this. It takes 2 minutes to scan these slides, and we're scanning in 3D, actually. And so the way it works is we have this full 9 by 6 centimeter area. This is what one microscope will see, this green patch. But remember, we're zoomed in. And so we don't see this whole green area. We just see a little blue square of it. And we need to scan in a 7 by 7 grid to fill in that green square. When we scan in a 7 by 7 grid, right, each microscope, all 48 of them are scanning. And so they're all going to scan out their respective squares. And so when we do a 7 by 7 scan, right, we capture 49 images from each microcamera, then we're done. We've covered the entire field of view and we can stitch all that data together. But it's a lot of data, right? Each scan is 31 gigapixels, and that's just in 2D. We're also scanning up and down in 3D. And so we create really large terapixel scale images with this type of technique. Here's just an example of scanning a well plate, right? Zooming in higher and higher to see this type of resolution. Let's see if this video plays. We have a viewer to show this type of data, but it is pretty hard to share or naturally show these type of tens of gigapixel images in a PowerPoint presentation. But this is just a summary of interacting with this data in our gigapixel viewer. We can image or scan in 3 dimensions, right? Naturally, to see these thicker specimens like fine needle aspiration smears. And so we capture, when we do a thick specimen like that FNA smear, something like 20 slices in Z, and we capture 0.62 teravoxels in that case, which is a lot of data. We're quickly synthesizing that down into all-in-focus composites, for example, for then pathologists to view and make assessments on. And then we're still using this technology to image these multi-well plates. And so a big application in that space is looking at organoids. So these are small collections of cells that are, created to model the behavior of individual organs in our body or in the body of animal models, which are naturally three-dimensional as well. Okay, and so I think I just have let's say 5 more minutes. I want to spend talking about the last topic. What else can we do with these multi-aperture systems? And so essentially, we've been making flat arrays, right? So you might notice, hey, you have this flat array and you're trying to observe something at high resolution. You'd probably want to curve your array to see it from multiple perspectives at a better kind of angular diversity, or achieve higher resolution potentially by curving your array. And so ideally, we would begin to be making systems drawn like this, where we have a curved array of microcameras, all examining a certain volume of interest. Unfortunately, it's really hard to fabricate something like this and achieve the data rates we're achieving. And I won't get into that in detail, but this is tough. Instead, we've found it easier to use our flat arrays and put a parabolic mirror beneath them. And so this mirror is shown here. It's an example. We have a few of these mirrors, but they're pretty inexpensive. They don't need to be very high precision optical elements necessarily. We naturally need to calibrate for each mirror, and so we digitally correct for various distortions introduced by the mirror, but essentially we image through the mirror and such that all of the cameras are focused at the foci of the mirror, where we place a specimen in a clear vial. You can see shown here on the left. The field of view, or sorry, volume of view of this parabolic mirror microscope is something like a centimeter cubed. And then we get pretty high resolution. So we're, again, in the couple micrometer per voxel resolution case here. And so our design, again, we are imaging through the mirror, and each of these colors corresponds to a unique microcamera. Each of the camera's image is shown on the right. So you can see we're getting all of these different perspectives of here a zebrafish larvae. So here you can see it's looking pretty much straight at you. Over here, it's the side, right? Totally different side views. And so this video data looks something like this, right? Just one example. And then we're putting all that together in a kind of tomographic image solver to create fully volumetric video. Again, this is fluorescent video of these freely moving model organisms at high resolution. So this video is captured of this zebrafish larvae freely swimming within this volume at, I think, 240 frames per second. And you can see pretty fine detail. We're seeing individual cells labeled. And you can see it swims out of the field of view, right? The water was a little higher than the volume that we're capturing. But each of those red dots is an individual cell. So we're getting towards cellular resolution in this type of freely moving case, but here with full volume 3D. And here's one more example. This is of a Drosophila larvae, just freely wiggling around, captured from all these unique perspectives. That's just one example. And then our composites, here's 2 of them interacting. We're beginning to analyze this type of data to observe these volumetric deformations at high speed in real time, which otherwise has been really, kind of impossible to capture to date, to capture this volumetric behaviors in a synchronous manner. Okay, and so with that, I'll stop. I'll take a little bit of time for questions. I naturally want to thank everyone who's been involved in this. So that includes our collaborators here at Duke, the Nauman Lab, and that especially for the earlier work, our first camera array was really created in collaboration with them. My lab, Kevin, who's now a postdoc at Berkeley, was really instrumental. And so same with Kong Hyun, but so everyone has been working really hard on this. And then Ramona Optics is a company that I co-founded, like I said, with Mark Harfouche. Here are other people involved in this work at Ramona, again, who are commercializing this technology. And then naturally thanking our funding sources. And with that, I'll stop. I can stop sharing, I guess, and answer some questions.